Thank you so much, Christian, for being here.
No, I take this one. Thank you very much, Jill, for the short introduction. Ladies and gentlemen, dear colleagues, dear members of the International Cannabis Research Group community, it's a pleasure to give you some opening remarks here for this conference. Normally, when people ask me to do this in my function as a dean, I'm coming with some warm wishes to meetings, and I do this quite often, and these are very general remarks. But here, with this topic, it's a little bit different, because cannabis research at Humboldt-University started in my group in 2020 with the first cannabis research class. So with some students, some of them are still here in the group, coming always back. That's nice to see, in different positions, and therefore it is a subject which is relatively close to my heart, and that's why I'm not giving you the conventional opening remarks, and saying I'm wishing you a successful conference. Of course I do this, I wish you a successful conference, but I would like to say a few more words, because I think that we have achieved already something in the last years, since the opening of this topic, it became possible to do research. For us, the research class started not only teaching students in a format, it is something where we were very interactive and where our interest has driven this research. Interest in a very complex plant, in an organism which offers so much, for horticulture, for pharmacy, for all kinds of research interests. And this was one part, and we have been driven right in the beginning to move beyond stigma and regulation because of this potential from this plant. So this slide sets the starting point of my talk. Cannabis science is entering a phase of maturity. For a long time, the field was shaped by fragmented observations, practical experience, and often vague strain of cultivar descriptions. Today, we are moving towards a more multidisciplinary, standardized, and evidence-based science. I think the first key step is genomic characterization. And we need to move away from these loose strain narratives towards clearly defined biological identities. Only when we know precisely which genetic material we are working with, we can compare results, production data, medical outcomes in a very meaningful way. The second step is controlled cultivation: light, temperature, nutrition, water supply, stress, harvest time. These are not only secondary factors. These are defined variables that can be measured, adjusted, and optimized. In controlled agriculture, controlled horticulture, cannabis production becomes a reproducible and scientifically meaningful process. Keywords here are AI-driven, climate control, dynamic light recipes, chemotype-oriented production systems, resource-efficient production systems, all subjects we are working on in the moment. The third point is particularly important for the medical sector: validated analytics and harmonized standards. So it is not only important that producers ensure high quality, that is kind of a given. It is equally important that physicians, pharmacists, and patients know exactly what they are receiving and in which quality. Medical trust depends on reliable information about cannabinoid profiles, metabolites, contaminants, stability, and batch-to-batch comparison possibilities. The image on the right illustrates this transition very clearly. Cannabis is no longer shown as an informal or poorly defined crop. It is a highly controlled, technology-driven production systems producing high-quality, uniform, well-described pharmaceuticals. The genome defines the potential of the plant. It determines what the plant is genetically capable of producing, for example, basics, capacity for cannabinoid, terpene, and secondary metabolite synthesis. But genetic potential alone is not enough. The actual chemical profile depends strongly on the environment, light intensity, spectrum, temperature, nutrient, water availability, very much stress, plant density, harvest timing. All of this is influencing how this potential is expressed. This is where ecophysiological knowledge becomes essential. The next step is metabolite synthesis. What matters medically is not only the plant name or the strain label, but the reproducible chemical phenotype. So cannabinoids, terpenes, other secondary metabolites in a defined and stable matrix. Finally, this chemical phenotype has clinical relevance. For medical cannabis, the patient and the physician need to know what is actually present in the product, in what concentration and what degree of reproducibility. So the key message is, genome tells us what is possible, cultivation determines what is expressed, and analytics make the results visible and trustworthy. For medical cannabis, it is essential that producers work under controlled and standardized conditions, but quality at the producer level alone is not enough. The question is whether this quality or quality information actually reaches the patient, the physician, and the pharmacy. Patients should know what they receive, not only the product name, but its actual quality, cannabinoid profile, terpene composition, safety parameters, batch identity, and so on. Physicians need defined and reliable profiles in order to make informed treatment decisions. Without this information, medical cannabis remains difficult to compare, difficult to prescribe, precise, and difficult to evaluate clinically. Pharmacies also need reliable batch information and traceable analytics. They are an important interface between producer, physician, and patient, and they must be able to trust the documentation behind each product. So the central message of this slide is transparency is not a marketing detail. In medical cannabis, transparency is a scientific and medical necessary element. If we want cannabis research and medical cannabis use to become more robust, we need a shared framework across disciplines, across countries. This framework should connect plant science, cultivation technology, analytical chemistry, digital documentation, and clinical practice. The first step is comparable plant material. We need clearly documented genomic identities, chemotype information, so that studies and products can actually be compared. Second, we need standardized cultivation metadata. Light, nutrients, stress conditions, harvest timing, post-harvest handling must be recorded systematically. Without this context, analytical results remain difficult to interpret. Third, we need validated analytical methods. Batch comparability, contaminant control, reproducible chemical profiling are essential for scientific credibility and patient safety. And the fourth point is digital quality passport. This means translating complex analytical and production data into accessible product information for physicians, pharmacies, and patients. And finally, we need a stronger clinical linkage. Chemical profiles must be connected to safety, efficiency, tolerability, and to be reproduced in data in a real-world medical use. So the message of this roadmap is clear. The next decade should not only be about producing more cannabis data, it should be about making cannabis data comparable, transparent, clinically meaningful, and useful for patients. This brings me to the concluding message of my talk. The future of medical cannabis will be defined, in my personal opinion, by two principles: predictability and transparency. Predictability starts with the genome. We need clearly defined plant material, reproducible genetics. It continues with cultivation, light, nutrients, temperature, and so on. And it ends with predictable chemistry, reliable profiles that can be reproduced from batch to batch. But predictability alone is not sufficient. It must be combined with transparency. The information generated during production and analysis has to be translated into clear, accessible documentation. Physicians, pharmacies, patients need to understand what a product contains, how it was produced, and how reliable quality actually is. This is the bridge from genome to metabolite profile and from production quality to end-user confidence. So my final message is medical cannabis will only gain full scientific and clinical trust if we connect predictable genetics with predictable cultivation, with predictable chemistry, transparent documentation, and medical relevance. Thank you very much for your attention.
Thank you so much, Christian. Maybe we can first open some questions to the room already. Anybody has specific questions?
Yes. With this, I mean, in order to do this, would it require a sort of certification that the strain or is, you know, it is what it says it is? I mean, how can that traceability be done to all the way to the patient?
I believe that we need kind of a quality certification, yes. I think that is something which should be reliable and which might be something which should be derived from universities which have not a direct interest in this, which are independent from companies and so on. I know that there will be a talk, and I think Daniel at least will talk also about cannabis transparency, and he might address this issue in his talk a little bit. There are groups working on that to ensure that we get this transparency idea of transparency, along the whole value chain.
Okay, and would that be something like added to the, let's say the microbial lab would do that extra checkup on that? Or how do you see that?
Can you repeat? I couldn't hear that.
Like, for example, on what stage do you foresee that happening? Like when it's already in flower stage where you can actually recognize it, or how could that be done?
No, this has to be done, of course, starting from the production, but the labeling and so on will be on the final product. The quality checkup will be on the final product, but to achieve this, you have to have standardization along the whole production chain.
Thank you.
I mean, not only when we talk about cannabis, the whole agriculture, horticultural industry is shifting more and more towards controlled environment systems. And this boosts our technologies, our opportunities. So when I mentioned a few keywords, because I know that everyone who has grown cannabis thinks, of course, I standardize, I know how to use light, and I have my light recipes, and so on. But we are making now the next step. We are getting more precise. We know much better, what happens in the plant when you change very simple factors, and of course, everybody has its own light recipe. There is not a unique one which works for everyone and for every strain and cultivar the same.
Yeah, kia ora. Paul Naskey is my name from Rural Bioscience in New Zealand. Just following up on this previous gentleman's question, you talk about validated test methods. I mean, maybe to put it in another way, I have a similar question. Like, what are you exactly and precisely talking about? Because we've got the EU pharmacopoeia, we've got COAs, which are all sort of standardized. What are you suggesting that gets added to that?
I'm not sure if I got everything. The acoustic is not the best in here.
Maybe my accent too.
No, well, I think it's more the acoustic. Let's phrase it that way. I think that's what I mentioned. We need someone independent, completely independent from the industry. We are working with different industries together, and they all claim, high quality products, and they all claim that everything which leaves their factory or their production system is perfect. And then we see at the end, what comes to the consumer is completely different than, what is on the data profile and on the chemical analysis, which has been done maybe weeks or months even before it reaches the final consumer. Therefore, I think we need someone independent, and we need a more flexible system doing analytics, testing really what is right now available in the pharmacy. What can you do right now, and what is the profile, so that you can really match this to real-world clinical studies?
Okay, so when you say profile, you mean maybe cannabinoids plus terpenes, plus other things?
Yeah, I mean, in the moment, of course, you know, a whole range of cannabinoids and terpenes, but we should not limit ourselves. I mean, when we talk about internal quality, it's more than that. It's more than secondary metabolites. We need to see that this is free of any other problems, you know, which we can see often in this crop, without going deeper.
Thank you. Any other questions in the room?
Hi, thanks. Could you clarify, are you talking about a standardization of the product and the metabolic profile of the product, as in, for this condition, there would be a matrix of the makeup of the plant, and whether that plant came from a company like Galaxia or another manufacturer, they would have to sort of hit certain criteria to get into that matrix for that condition? Or are you looking at it more as a scheme where, you know, we have GMP, but there's another thing on top of that whereby we're proving how consistent our production is of a strain within, so the consistency of that strain rather than the consistency of hitting the matrix of the sort of metabolic profile of the plants? I'm not quite sure which way around you think it's going to go.
I mean, GMP ends with the producer. And what happened after it leaves your production unit? You know, we are talking about medical cannabis here, or I was talking about medical cannabis, and there is a certain demand for that, you know. And people, I mean, in my family, I have people who get this prescribed, and we say, well, yesterday I got something which really worked. And, you know, last week I didn't get anything. It didn't work at all. Why? Is this the case? You know, and when I go and I just ask my MD, you know, to give me a prescription for aspirin, he gives me a prescription for aspirin and he tells me exactly how much I should take and so on. And this is very precise. And with a natural product where we have hundreds of different cultivars and strains, which have completely different profiles, you know, where we look only at very few numbers in this profile when it leaves your GMP factory. There is a huge gap at the end. And so we need something which is more flexible and which is more accurate at the time when the product gets to the consumer, to the patient.
So would a sort of a brand scheme, so, you know, in food in the UK where I come from, we have a red tractor symbol that goes onto food to say it's been grown to a certain standard. Now, GMP is only a measurement of safety. It's not a measurement of the genomic makeup of the plants. But if you had some sort of standard that said this cultivator and this process and this whole supply chain has met a certain standard, that means that the consistency of the product of this brand is within these parameters, so you know as a patient that if you get it 1 month, it's going to be much more similar to the one you get next month.
Yes.
Yeah, okay, understand, thank you.
This is a risk, you know, when I do opening remarks without saying, hey, welcome to Humboldt-University.
Hi. It's maybe a little bit a philosophic question. We are talking all the time about this metabolic profiling and that we want to increase it and get more numbers. But at the end, we would maybe have a list of 10,000 hits or 5,000 hits we want to compare and maybe give them the list to the physician and say, hey, interpret this. Is this the right medicine for your patient? I think at the end, it gets far too complicated if we include everything into our, I say now, COA, what we then want to give to the physician. So shouldn't we make it even not more simple, that we can better interpret it for the final step when it reaches the patient?
I like that question, to be honest, because I mean, I study biology and I keep saying natural scientists are very simple people because and in horticulture the same, you know, we make everything so simple because we don't understand complexity. At the same time, of course, here we have a unique chance to make kind of a reversed engineered research approach. So we use real world data, we use patient data, we use people who explain us what are their problems and what helped again with their problems, and then we get easily giant number of data which we can utilize to become much closer, but it's still a natural product, and there will be never the same, twice. So there will be always minimal changes. So we also have to make it easier, and we will not look into all 140 cannabinoids. We might focus in the beginning on 5, 6, 7, 8, 9, whatever, depending a little bit on for what it gets prescribed.
Yeah, it should be actually at the end a little bit in between the aspirin, with just one compound, and something it's a natural compound or composition of something.
I mean, this is not the first natural product I'm working with. So we did a lot with salicylates. We know that aspirin is on the market, which is only one active ingredient, and people are getting prescribed aspirin. And the natural product are salicylates. It's a big bunch of salicylates which comes from willow, from the bark of willow. So we are now producing standardized willow for the pharmaceutical industry in southern Germany. After a long breeding program, and we came up with only 3 willows, 3 chemotypes, which we now understand. But willow is having a higher variability because we are producing willow outside on the field. So every year it's different, but the basic chemotype is fixed.
Yeah.
That's the breeding part.
And you standardize then just on a few compounds in the mixture and say, okay, this has to be, I say now 100%, whatever.
Exactly.
Yeah, okay. Yeah. Thanks a lot.
Okay, maybe I will ask one last question to round off. We've been zooming in quite a lot on questions related to transparency. I would like to also focus a little bit on your role here at one of the most important research faculties in Germany. Germany has become a kind of a leader or a pioneer within Europe, at least on the medical cannabis field. What is the role that you think Germany can also hold on both the research part or this creating these transparent supply chains, etc., for the more European framework that could be put in place?
That goes a little bit too deep into politics, but I personally believe that since we are so much in the center of this and that we are importing in the moment quite a lot, that this is also our responsibility to take maybe the lead, to bring people together, to talk about this and to exchange the ideas. And when I mentioned standardization along, production systems and so on, this was intended to compare research, to see what is going on. In the moment, we are working with different groups in different countries, and it's difficult, to come up to the same conclusions. We try to come closer to understand what's going on in the plant because the systems are so different. So we need to standardize here.
Okay, thank you so much.
Thank you very much.